This video is going to be all about B cell activation, which is, again, B cells are part of our humoral response, which is also called antibody-mediated immune response. So remember, we've been talking about our specific immunity, and there are two branches or two arms to our specific immunity, and that is going to be the humoral response and the cellular response. And so what we're going to talk about now is how we activate this humoral response so we can eventually mass produce antibodies that we need to clear an infection. Now, what we're going to do is walk through the antibody-mediated immune response. And so there's a diagram online that you will want to have handy. And there's a second one called the phagocytosis diagram that you might want to have handy as well. And so with this video, we're going to go kind of still image by still image to make sure you understand what you're looking at on these handouts. So again, the big idea is antibody-mediated immune response, but we also call that B cell activation. And B cell activation is normally what I'm going to call it, just because that's the goal of this process, is to activate our B cells so we can eventually make plasma cells and make some antibodies. All right, so this comes from the antibody-mediated immune response diagram. And essentially what is going to happen is when there's maybe a break in the skin, right, microbes get in, some scary pointy looking bacteria here. Notice the little triangle structures on the end are going to be the antigen, and that the antigen is going to be what is detected by the antibodies. And so notice here we have a dendritic cell, which is an antigen presenting cell. So you'll notice it already has its MHC2 marker displayed. Remember, you only find MHC2s on antigen presenting cells, but remember they also have MHC1s because they are self-immune cells. So the dendritic cell, in order to present antigen, it has to first be able to phagocytose the bacteria, because by ingesting it and digesting it, it can then take part of that digested material and present it, okay? And so notice here on the left, you can see where it has ingested or is in the process of ingesting the bacterium. It is going to go the phagocytosis process, which we'll detail more here on the next slide. But then once it's phagocytosed the bacterium, notice what you see on the right-hand side of the dendritic cell. Now, part of that bacterium, that antigen, is now associated with that MHC2 marker. Okay, and so that's going to be really, really important. That's the end goal here. Okay, so let's review phagocytosis really quick so that way you can understand how it's able to present just the antigen portion of the bacteria. So remember, first the phagocyte is going to go and hunt down the bacteria via chemotaxis, and then it is going to attach to the bacteria. So remember, the pathogen has what are called PAMPs on the surface, which are little molecules that will bind to the TLRs on the phagocyte. Once it attaches, it is going to do endocytosis, and it is going to take the bacteria into its own little vacuole. We call this the phagosome. Remember, the phagosome is going to have some proton pumps to be decreasing that pH, because look at what happens next. That lysosome comes and fuses with that vacuole. We now call this the phagolysosome, and little hydrolytic enzymes that are in the lysosome are going to chop up that bacteria. So here you see in this residual body, this third little vacuole, notice that the bacterium is digested. Okay, it's been chopped up into pieces, but that surface antigen is still intact. That's just one of the digested pieces. Okay, so what's going to happen next is inside this dendritic cell, okay, it's got a little Golgi here, And coming from the ER and the Golgi are going to be these little MHC2 markers that are packaged into their own little vesicle here. And so what's going to happen is we've got on the right here that little residual body with all the digested pieces, including the antigen, the triangle. We also have these little MHC2 markers that are now in their own little vacuole, and they are going to merge together, okay? And inside this little vacuole or vesicle, whichever you want to call it, the antigen is going to bind to the MHC marker. Okay, so now we call that an MHC antigen complex because they are associated with one another. So remember, this vesicle is going to leave the cell via exocytosis. And from there, the antigen MHC complex will be inserted into the plasma membrane of the dendritic cell. Okay, everything else is going to be released outside of the cell because it's just trash. Okay, and so now because the antigen is bound to that MHC2 molecule, which goes to the plasma membrane of the dendritic cell, now that antigen is being presented to other white blood cells. And so they function as a wanted poster, right? If we're keeping this PG, it's allowing the white blood cells to know that, hey, I found this pathogen. I've eliminated this one, but I bet there are others. So this is what you're looking for. And it's displaying that antigen. Again, I'm a Game of Thrones kind of girl. And so I think of like the head on a spike, right? If you haven't watched Game of Thrones, it's pretty good. But some pretty likable characters are killed off in the first season, and their head is displayed up on spikes, right? Because that lets other traitors, they say, know that, hey, we found your little friends here and we're coming for you. And that's essentially what the antigen MHC complex is doing. It's alerting the other white blood cells that there's a problem and we need to go hunt them down. So here's some things that I just want you to think about. So if the dendritic cell presented as antigen with no marker, okay, so we're going to think about that as we continue on. And APCs present complexes on their surfaces. Who are the APCs? We've already answered that, so go back and look through your notes. All right, so some critical thinking questions that I want you to ponder. We can revisit them at an exam review. So far, we've talked about how a dendritic cell has identified a pathogen, digested it, and presented the antigen on its surface. So that's kind of where we left off here on the left, okay? So you can see the antigen MHC complex is presented, and notice here we have a virgin T cell, okay? And T cells are part of the cellular response, and T cells can only recognize digested antigen, also called antigenic peptides, okay? So what's gonna happen is this virgin T cell here has a receptor that is going to recognize this antigen MHC2 that's on the dendritic cell, okay? And it's able to bind it, which is what you see happening here on the right. And so once that binding happens, the dendritic cell is going to release little molecules called interleukins, also called cytokines, that are going to induce the T cell to divide into different types of T cells. We're going to have helper T cells, memory T cells, and cytotoxic T cells. It's the T cells that make cytokines, my apologies. So the T cells are going to produce their own interleukins or cytokines, and we're going to talk more about memory T cells later when we get to our secondary responses. But all of these T cells that are made are going to have that same complex receptor that the original T cell had, because it is specific for that pathogen that the dendritic cell had phagocytosed. Okay, so let's kind of put all of this back together, okay? So again, there's a break in the skin, the microbe has gotten inside, we're going to call it a bacteria for this example. The dendritic cell here is going to phagocytose the bacteria, okay? And this is going to cause the release of interleukins by dendritic cells, which is basically how they communicate with one another. After phagocytosis is complete, right? Remember, the antigen will be present inside the dendritic cell. It's going to fuse with a vesicle that has the MHC2 marker. And so then collectively, the antigen MHC2 complex is now presented on the surface of the dendritic cell. And so this is what a T cell can recognize. It can only recognize digested antigen. And so this little virgin T cell is going to bind to that antigen MHC2 complex on the dendritic cell. And in doing so, it releases its own cytokines, and the T cell is going to divide into various types of T cells. And what we're concerned about for this process, which is B cell activation, is we are worried about T helper cells, because T helper cells will help activate the B cells. Okay, so the two arms of specific immunity are connected to each other. In order for B cells, which are part of the humoral response, to become activated, they need to be helped by a T helper cell, okay, which is part of the cellular response. All right, so all of this has happened. Okay, we've activated our T helper cells, but you might be thinking, Chelsea, you said this was all about B cells. Where are the B cells? Okay, well, we're getting to that. But remember, they need the help of T helper cells. So I wanted you to understand how those got activated. Okay, so we're still in the same event has happened. Skin is broken, microbe has gotten in. Well, remember, on the surface of B cells are tons and tons and tons of antibodies that are specific for that unique pathogen, and they're essentially circulating through trying to find its pathogen. Okay, so that's our little virgin B cell with all of its antibodies on the surface. And eventually, this B cell is like, hey, wait a minute, you're my antigen. I am specific for you. And so they bind together. And B cells are also antigen presenting cells, and they are able to digest intact antigen, right? Because check it out, it recognized, right, this virgin B cell recognized the intact, complete, unprocessed pathogen. It then phagocytoses it and digests it. And just like the dendritic cell that we just talked about, it creates an antigen MHC2 complex. Okay, it's an antigen presenting cell. It functions very similar to the dendritic cell. So now the B cell is considered activated because it now has that antigen MHC2 complex. So now that we have our activated B cell, notice here comes that little T helper cell that the dendritic cell had activated. So the T helper cell, T cells recognize digested antigen, which is now on the surface of the B cell. So that T helper cell is going to come in and bind to the B cell, which is going to allow this B cell to divide. Because remember, T cells produce cytokines, also called interleukins. And those cytokines that the helper cell releases are going to be what activates that B cell to divide. And notice that that activated B cell, as it divides, it differentiates into three different types. Actually, on this picture, sorry, I'm getting ahead of myself. We're only showing the plasma cells, but there are different types of B cells. So our plasma B cells are going to be secreting, notice, the three different types of antibody that we talked about previously. We have the monomeric antibody, we have the dimeric antibody, and our pentameric antibody. Okay, and so these are mass-producing antibodies that are going to be able get into body fluids and all of the areas that white blood cells can't go in our body. And these antibodies are going to perform various functions that we're going to talk about in a future video, but they are listed here: neutralization, opsonization, complement, fixation, and mobilization. And neutralization was already said. Now, plasma cells can switch antibody classes. Remember, we talked about class G, A, M, E, and D. And so by switching classes, that's how the different forms of antibody are released, right? That's how we get the monomeric, dimeric, and pentameric antibodies that you see here, okay? But remember, no matter if it's a monomer, dimer, or pentamer, they all have that same paratope because they're all going after that same antigen that original virgin B cell detected, okay? All right, so that is B cell activation in a very, very large nutshell. So let me real quick just kind of recap it without any pictures. Okay, so just kind of listen to my voice here more in a stepwise fashion. So a inactive or virgin B cell is going to be covered with lots of antibody. And that antibody is specific for a pathogen. And so when it first encounters that pathogen, it will phagocytose that entire intact pathogen. It will form the antigen MHC2 complex that will be displayed on the surface of that B cell. That B cell is now considered activated, but it can't actually differentiate into plasma cells until it is kind of activated a second time, but by a T helper cell. So that's why we need T helper cells to become activated. Well, dendritic cells play a role in that. Okay, so the dendritic cells are also coming into contact with that antigen, phagocytosing it, presenting the antigen on its surface. That's going to be what the T helper cell binds to, and it's going to, the virgin T cell, the inactive T cell, going to bind to that dendritic cell that is presenting the antigen that is going to activate it into a T helper cell. So now we have an activated T helper cell and we have an activated B cell. And what we need are antibodies, okay? And so that T helper cell is now going to go and interact with that antigen MHC2 complex that the B cell is presenting. And in doing so, it is going to release cytokines that are going to trigger that activated B cell to divide into plasma cells. Remember, plasma cells are specifically what releases antibodies. And it's going to be releasing the different classes of antibodies. That way we can get as much protection as possible. All right, y'all, I know this is a lot to digest. So watch this video. I posted a YouTube video as well. Really give yourself time to kind of go through this because you want to know this diagram. So both the antibody-mediated response diagram and the phagocytosis diagram for the exam. You need to be able to explain this process on your own, kind of like the recap that I just did, in order to make sure you really fully understand this process come exam day. All right, y'all, that is going to be the end of this video. Please let me know if you have any questions.